So the structure of axon that we can see if we make a cross section of it, so the interior of the axon, which is called the intracellular fluid matrix that is present there, in this ICF region, it is usually noticed that within the ICF region there is a large quantity of phosphate ions. There are negatively charged proteins that are there. There are a large amount of like potassium ion that can be found present in this ICF region. Okay, the most of these ions actually they build up in the interior, this interior part of this membrane, which is called the axolemma. Okay, let me write the name here, axolemma. So it actually is like present within this membrane region of this axolemma. And in contrast to this, in contrast to the intracellular fluid, the extracellular fluid matrix, which is just the outside portion of the of the axon, we can say that that area is it has a buildup of a very large amount of sodium ion and a very large buildup of chlorine ion. Okay, now sorry, now that buildup actually creates a positive charge on the outer surface, and in contrast to all of this, there is buildup of negative charge on the inner surface. Okay, because this two charge difference, it is completely dependent on what kind of ions are present there. Now sodium is present in a bit higher quantity there with comparison to that of the potassium ion. It what actually creates this charge differentiation in the in the entire membrane. Okay, now what we are going to find here is that this situation which is present, which can be found in the axon, is known as the resting potential state of the this axon. Okay, now during this resting potential state, we have to keep in mind the sodium always will have a tendency to move inward, and the potassium will have a tendency to move outward, because potassium will is always like it has a capacity, or it has a ability, or a property, we can say simply, that it always wants to travel through the membrane outward. Okay, means the other side of the membrane. Whereas the sodium, it has a less property of moving inward or something. Okay, now what actually happen is this potassium trying to move outward, you may say that it will create even more positive charge, but this process is completely stopped by the presence of these phosphate ions and the proteins ion that are present there. Okay, now phosphate and protein being a very large structure comparison to that of the potassium and the sodium, which are simply a single ion of a single atom, isn't it? So in the atom, this is in the atomic state, this is in a comparatively large molecule, and this is the definitely a very large molecule, right? So all these molecules, they are not able to move outward, right? Means these proteins and phosphate, they cannot move that outside that easily, whereas this potassium can easily travel outward. But the sodium, on the other hand, also can travel inside, but the sodium has got a problem with the chlorine, which is trying to pull the or keep the sodium in that particular place only. Plus, sodium cannot travel inside the cell that easily. Okay, it requires some sort of means transport proteins, so that the transport protein can actually bring sodium back inside. Okay, now what actually happened is, at the time of like this generation of this action potential, now what we actually will find that there are specific proteins that are present in the in the cell. Okay, means in this axon membrane, which is responsible for transport of the ions or exchange of the ions across. Now that particular protein is known as a sodium potassium pump. Okay, That one is known as a sodium potassium pump. Now sodium potassium pump, it is just a structure which is like I can simply, it's like we can make like a simple diagram with this. Okay, now it is like a structure which is like slightly we can make it like this way simply. Okay, now this molecule, this particular structure, it has the property of transport of this ions across. Okay, transport of the ion across, and for that it is mostly going to be like it's mostly going to be with the help of ATP, expense of ATP. Okay, now ATP or energy mediated transport is what is known as the active transport. Now with the expense of ATP, we are going to find that it can actually move 3 sodium ions, okay, in this direction, and 2 potassium ions in this direction. Means the movement of ion will be in opposite direction to each other. Okay, it will be in opposite direction to each other. Now usually this is done with the expense of utilization of adenosine triphosphate, means utilization of energy. Now the sodium potassium pump is what is going to act as the main driving force for changing of the charge or altering the charge of the membrane. Because if we find that the sodium is moving, let's say from the ECF region to the ICF region, then the positive charge that was present initially on top will actually now alter, because now in the outside surface we are going to find there are less amount of potassium outside. Okay, because it will not allow 3 potassium to move out, but rather only 2. On the other hand, 3 sodium is moving inside. Now what is happening is this excess of the sodium inside will actually create a positive charge in the surface of the ICF inward region. Okay, so we are going to find that due to this change, due to this altered altercation that is made by this protein, say this is the protein. Okay, so the altercation that is made by this protein is actually allowing the movement of 3 sodium ion inside, and it is making 2 potassium ion to move outward, but that one will create a temporary negative charge onto this particular surface, whereas this side it will become positive. Okay, from the previous state of being outside positive and inward negative, it has completely altered to a different conformation. This altercation of the charge is what is known as the depolarization. Okay, and this depolarization, it is all going to be mediated with the expense of ATP, but this depolarization will actually trigger the adjacent side, means the adjacent side, whatever see, the rest of the thing is actually in the previous state. Now it is in the stage of like that normal condition of outside being positive and inside being negative actually. Okay, so this portion, since it has got this change in the charging state, okay, this depolarization occurring in one of the side, and that depolarization will now influence this other side. Okay, because the sodium potassium pump here is actually not only this like active type of or mechanical type of sodium potassium pump, there are also varieties of them present there, of which this variety that is present here is known as, this particular variety of them is known as the voltage gated pump. Okay, the voltage gated pump means the moment it sees this or this altering of the charges on the other surface will actually create, this is a creation of the action potential which will create this or induce this particular protein to go for this exchange of the charges. Means now it will force 3 sodium to move outward. This 3 sodium that has moved inside, it will force the 3 sodium to move outside, sorry, and 2 potassium to move inside now. So that is what going to happen now. Okay, that it will now force sorry, I forgot to draw this one. So this depolarization will now trigger this other molecule to first of all, though, this is going to happen, sorry, first of all, though, is again sodium is going to push inside, okay, and potassium is going to move outside. Means whatever sodium is there, it will be pushed inside. Okay, sodium, 3 sodium moving inward now, and 2 potassium moving outward now. Okay, so that means now this side will become negative and this side will become positive in comparison to this whatever was present there. Okay, means it was initially negative here. Okay, now this negative charge, sorry, this was like positive and negative on the other side, like oppositely. Now this side will also become the opposite, means this side is now getting depolarized. Okay, but that depolarization, when this side will become depolarized, at that very moment we are going to find that this side will actually bring itself back to the previous state. Okay, with the expense of adenosine triphosphate into adenosine diphosphate and pi. Okay, it is going to bring itself back to the previous state with 3 sodium being pushed outside and 2 potassium brought back in. Now that will create that previous state of positive charge outside in negative charge inside. That particular state will be created. Okay, so that means we have this side repolarized. Okay, so let me tell you what is happening here again. So first of all, the cell, first of all, this entire thing is dependent on the axon. So the axon, the moment it receives the stimulus, and then it has initiated the action potential, or in which it will force this charge differentiation to take place in the cell. So the moment this is generated, with that very moment the sodium 3 sodium ion will be brought inside and 2 potassium ion will be brought will be taken outside. Okay, so this ECF region and ICF, I forgot to write that. Okay, now that will actually create differentiation of the charge on the surface with whatever it was there present previously. It will create a differentiation of the charges on the surface. Okay, the top will become, the outer side will become now negative and the inner side now become positive, and that will trigger the adjacent voltage gated pump to repeat the entire process the same way. So thus we are going to find that this side will actually bring the sodium inside now, and it will send 2 potassium outside from this region. Exchange will take place. Okay, and while that is taking place, means this depolarization which occurred here, the same depolarization will now take place in the number 2 position now. So that depolarization, when it is taking place, with the expense of ATP, the previous one will actually bring itself back to the normal state, where it is like repolarized back to the previous state of whatever charge was present, means outside positive, inside negative. So we can see the depolarization has moved one unit forward. If we consider this a unit, then it has moved 1 unit forward, and that is going to continue. If we draw it again, means if I am to show you with the progressive diagram, then we can say that now the same thing is going to move ahead with the next unit. Okay, let's say here is another unit. So here it was positive, here it was negative. Now we are going to find that here it will turn negative, here it will turn positive. Why? Because sodium has moved inside and potassium went outside. Okay, same thing is going to happen. And when that is happening, at that particular moment, we are going to find that sodium being sent outside, okay, and the potassium was actually brought back inside in this particular side. So what we will find that this area will now again become positive in the outer side and negative on the inner side. Okay, it will become positive on the outer side and negative in the inner side, but this area has got depolarized. So we can notice here that the depolarization is moving forward, isn't it? We can see that the depolarization is moving forward. If we even want to shade it slightly, let's put a little bit of shade, then we can see that the depolarization is now moving forward, isn't it? And that is what is happening inside the with the axon. Okay, that is how the charge is getting moved from one part to the next, 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 and ultimately will reach the synaptic terminal or the end of the axon, and where that will trigger the synaptic vesicle to fuse with the wall and finally release the neurotransmitter, which will be accepted by the adjacent neuron. Okay, so that is how the electrical signal is conducted via the membrane of the axon, because it was mentioned that in case of the axon, the entire axon, it actually runs by an electrical signal. So this is what that electrical signal is all about. This is how it is transported. Okay, thank you.